David, thanks very much for inviting me. During my years in Jerusalem, I learned a lot from you, and not only about physics and um, statistical mechanics, but also the fine points of Jewish law. I still remember the instructions about Ben Hashmashot. So I'm also honored to be giving a talk in honor of Carl van Vreeswijk. He was really a fantastic scientist and a real gentleman, a really nice guy. And so it's really great to be honoring him this way. All right, so can everyone see my screen? Great. So these are two special issues of Nature that came out. They're the products of two consortia that I was a part of. One of them on the left was about the fly connectome, the wiring diagram of the whole fly brain. And the right-hand side was about a little tiny piece of mouse cortex. I'm only going to talk about the left-hand side first in this talk. So the FlyWire Consortium was organized by me and my colleague Malamurthy at Princeton University. It ultimately grew to include 75 laboratories and a bunch of citizen scientists. And I think it's safe to say that all these labs are publishing lots of papers and making discoveries. They're really finding the fly connectome to be useful. I'm not going to talk about how we got the fly connectome, really. In fact, there was a race. We got there before Howard Hughes Janelia in a story that revolves around personalities, technology, politics, and simply luck. It's strangely enough, the fly connectome that we reconstructed was based on images that were originally acquired at Janelia. But they actually abandoned those images and decided to take other images. And so that's how we were able to get to the fly connectome before Janelia, but of course, in the end, using data acquired by Janelia. So it's with Janelia in that sense. All right, I'm not going to talk about that. You know, I would like to say though that the fruit fly brain did capture people's imagination. We did get 15 seconds shout out on Saturday Night Live. Um, But really the question is, so what, right? And, suppose you have a connectome, is it really useful for science? And I want to remind you that over a dozen years ago, there were plenty of famous people really questioning that idea. So here's a quote from Tony Mobshawn in 2012. Um, Our understanding of the worm has not been materially enhanced by having that connectome, blah, 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 blah. Um, But that connectome by itself has not explained anything. Um, I think that Eve Martyr was more pithy, very concise. She said connectomes are absolutely necessary and completely insufficient for understanding nervous system function. So yeah, necessary and not sufficient. That's one thing to say, but she went further and said completely insufficient. And so this really seems kind of negative. And sometimes people ask me, why were these famous scientists so negative? And my interpretation is that it's really about their pessimism was really about, well, even if you have a connectome, it might be really, really hard to understand. And you can take the analogy with the genome, right? So suppose you have the sequence of a gene. Does that tell you what that gene does? Amazingly, it's very atheoretical. What do? You don't really read that gene, you just knock it out. And then you see what happens. What actual biological function does it affect? Of course, later on, you can, if a similar gene is found, then you can guess that it might have a similar function to that earlier gene with a similar sequence. But the first time that you ever see a sequence, it's almost completely atheoretical. You just have to knock it out, very primitive way of thinking. And so in some sense, you could say that the DNA sequence is really inscrutable. It's not comprehensible. And so maybe Eve and Tony were really skeptical about the idea that the connectome would be interpretable. Maybe they thought, of course, yeah, you have to have the connectome, but it's just going to be really, really hard to interpret afterwards. And so really that's the question that we've been confronting with the fly connectome. And so what I'd like to argue today is, actually one of the most surprising things that I found is that the fly connectome is surprisingly interpretable, at least surprisingly to me. And so I'm going to really discuss the visual system of the fly in particular. So as a corollary, if you have the entire fly brain mapped out, you also have, it's the first time that an entire visual system has been mapped out at the synaptic level. And so this paper was spearheaded by Ari Matzlia and CJU. We went and we actually took almost 40,000 neurons in one optic lobe of the fly, and then we classified them into types. And let me just remind you that there's really historically many approaches to cell types. The first one, which we often credit to Cajal, is just to look at the shapes of cell types. So here's a old paper by Cajal and Sanchez on the visual system of a fly, and some of the cell types can be recognized from their drawings. And then later on, and more recently, for example, at Genelia, there was a combination of molecular and morphological cell typing. So take a transgenic line in which a few cell types are visible in the fly brain. And then now that you've restricted the cells to those few cell types, you can use morphology to distinguish between them. And then more recently, all the rage has been transcriptomic cell typing. So you look at the gene expression in neurons, and then you try to cluster that. And what I want to tell you about here is connectomic cell typing. So what does that mean? Well, we have a feature vector for every cell, but it's not gene expression. It's the connectivity with other types. So let's say there's 200 plus cell types that are intrinsic to the optic lobe. Each of these feature vectors, there's 3 cells here, and each feature vector, each feature vector is just the number of synapses received by this cell from each of 200 plus types, and the number of synapses sent by this cell to each of 200 plus types. So it's a 400 plus dimensional vector. And you can see that the feature vectors for cell 1 and cell 2 are more similar to each other than they are to the feature vector of cell 3. And in fact, cell 1 and cell 2 are part of the same cell type, whereas cell 3 is a different cell type, but in the same family, this so-called PM family of cell types. So this feature vector is a very rich representation, and it turns out that it's very powerful. And we've identified all of the cell types in the optic lobe. We more than doubled the number of known cell types that were found by these other previous methods. All right. And it may sound a little circular because I have to have the cell types to define this feature vector. But what you do is you just start out with morphological cell types, old-fashioned cell types. And that's good enough to make a feature vector that allows you to split and merge and then revise this representation until you actually get the real cell types. And it's self-consistent. So once you have this representation, the clusters are tight, they're compact, and they're well separated from each other. So I would say though that the algorithmic problem of finding a clustering algorithm that just starts from the connectivity matrix is still an open problem that some of you might want to tackle. All right, so we've added new cell type families. The cell type families are classically defined by their projections in these 4 major neuropils of the optic lobe. So you can see the 4 neuropils cartoon right here. And in fact, this new SM type right here is hugely, it already has 50 new types. It's a huge number, huge cell type diversity in this SM family, which was not known before. And then you can see here, this is a graph of the number of cells that we've recovered from every type. And you can see that the most numerous cells, the cell types, some cell types have 800 cells in them. And they are the ones that cover all of the pixels of the fly's compound eye. Because in this fly, there's about 800 ommatidia inside the compound eye. But there's plenty of cell types that are fewer than 800. And in fact, there's many cell types for which there's only one per optic lobe, one giant cell that covers the entire optic lobe. And so, and as I said before, we have more than double the number of known cell types. All right, and not only that, Howard Hughes Janelia, 6 months later, they published their own optic lobe. They still haven't published the connectome yet. But they found almost exactly the same set of 200 plus cell types intrinsic to the optic lobe. And the only differences are probably a few cell types that are either male specific or female specific. And so this is a incredible replication. Most cell typing efforts you'll find different investigators can't agree on the cell types. And one big issue in cell typing is this lumper splitter dichotomy. Here's Charles Darwin writing about the lumper splitter problem in taxonomy. And the reason why we're not affected by that, or we're not really badly affected by that, is that we have a well-defined way of stopping the splitting in the visual system. Because most cell types, almost all intrinsic cell types in the visual system, need to cover the entire visual field. So we keep on splitting, but if now we split into two groups that fail to cover the entire visual field, that's going too far, right? So that's how we know when to stop. That's how the lumper splitter thing is not subjective. It really has a more objective criterion that of where you actually have to set the coarseness or fineness of the clustering. Okay, and I should say that the clusters themselves, their definition, remember the feature vector doesn't have any information about space in it. So when you get a nice coverage, that's actually kind of validation of the whole method, independent validation. Okay, and then and I just want to make a point here about the unreasonable effectiveness of anatomy. What we found is that some connectomic types cannot be distinguished by morphology. So they look the same, but they put their arbors into the same locations inside the brain, but they actually connect to different targets inside their arbors. So that's why they can't be distinguished by morphology. It's also been found by the transcriptomic people that some cell types are transcriptionally indistinguishable in adults. Here's a great example of the transcriptomic clustering of the motion-sensitive neurons in the fly visual system. And you can see that the clusters are best halfway through the pupa stage, 50% of the 50 hours through the pupa stage. But then when the fly becomes an adult, those clusters start to merge again. And this is something that the old-time anatomists always said, if the transcriptional differences are there to establish wiring patterns, they might only be there during development. There's no reason why they have to be preserved in adulthood. And then a third point is that transcriptomics currently stumbles on cell types with low cardinality. And you saw that there's plenty of cell types with only 1 cell per type or 10 cells per type or 50 cells per type. And transcriptomics currently these transcriptomic researchers haven't been able to pull those out. All right, so anatomy is really the gold standard here. All right, so that's the cell types, but now we want to move on to the question of function. And so, here's where people are often skeptical. Can we really learn about the mind of a fly by mapping a dead brain, right? People are often very skeptical about that. And I'm going to quote somebody else. Here's Marcel Masalem, who is an expert in human brain anatomy and function. And his quote is, his soundbite is, "Nothing defines the function of a neuron better than its connections." And so those feature vectors, which we took as signatures of cells, we could also take them as signatures of cell types. Just average the feature vectors together for each cell type. And then you can make a family tree. So here's all of the 200 plus cell types. And we are computing an average feature vector for each of them. And then we are computing their relatedness by the similarity of those feature vectors. So these two cell types, it's a hierarchical clustering here. These two cell types are close together. So they're siblings, very close siblings. And then these groups of cell types, they would be distant cousins. And so one of the beautiful things that comes out of this is that this hierarchy actually makes sense functionally. So I want to emphasize that less than 20% of these cell types has ever been recorded from by a neurophysiologist, right? So we have a sport here now, which is, can we guess the functions of the other 80% before they're measured from by neurophysiologists, right? But what we can say is that if we just look at the ones that have been measured, they look sensible here. For example, here's T4 and T5 are all closely related by connectivity. And in fact, they all have similar functions. They all are detecting, they're the motion detectors for detecting motion in the 4 different directions for light and dark stimuli. So indeed, they are close together in this hierarchy. There are, on and off channels. There's small object detectors like T3 and T2, and they happen to be close together in this hierarchy. There's cell types that detect polarization, cell types that detect color, spectral sensitivity. They're close together in this family tree. So even though no information about physiology was put into this family tree, only connectivity, we end up with something that looks interpretable in terms of function. And so we actually hypothesized, this is very speculative, but just based on this diagram, we hypothesized that this is a motion system, this is the color system, this is an object system, so we don't know if these are all hypotheses already coming out of the connectome. Now, you could say, hey, well, that's not, that's still more like the genome, right? It's very atheoretical. You just look for similarity in the connectivity, and then there's some neurophysiologist who recorded the function, and so you're not really using the connectivity in a deep way to figure out what the function is. So just as an exercise, let's look at this part of the hierarchy up here. This is 6 cell types. They're very closely related, and until recently, none of them had ever been recorded from. And so we can't use the similarity trick to guess its function. We actually have to look at the connectivity, to do a deeper dive and see if we can guess visual function from the detailed structure of the connections, including their spatial organization. All right, so let's try to guess the functions of these cell types just for fun. And I want to emphasize that this is interpretation, right? So I want to emphasize that other people like Srini Turaga and Phil Hsu are working hard on realistic brain simulations. I'm not going to show this video, I guess, but they're working hard on simulations. And many of you have done modeling like this before, but you can make connectome-based models. But what I'm going to show you really is more like analysis and trying to predict the function without a simulation.
It's taken over a decade.
Okay, so here's a paper predicting visual function of these cell types. And I'm going to summarize what I found. And so first of all, the DM3 cells were first reported in 1969 by a famous neuroanatomist named Nick Strausfeld. And until recently, no recordings of them were published during, more than 50 years. And I'll show you what they look like. Oh, actually, before I show you that, let me just give you an idea of, this is our optic load catalog. I told you there's 200 plus cell types. Here is this catalog that you could find. This is the FlyWire Codex made by Ari Matzlia in Molly Murty's lab. And you can see this very large number of cell types. All right. And as I said before, less than 20% have ever been recorded from. And I mentioned that there are some cell types that are about 800 in number. For example, this is L2. And I guess not all of them are showing up here. I need to set my memory to be higher to show them all. But if they were all shown, they would be densely packed. And you can see that they're making a retinotopic map, right? They're receiving input from photoreceptors, and then they're making a retinotopic map in the medulla. Right, so these are sort of pixel types. Okay, so let's see if we can find the DM3 cells. I'm only going to show you 50 of them chosen at random. So, and here they are one at a time. So you can see that they look like snakes rather than trees. And here's all 50 of them together. So if you look at them carefully, you could notice that there seems to be some variation that is discrete rather than continuous. All right, so I'm telling you, here's a random sample of 50. But actually, these three cell types, these DM3 cells consist of three types. And it's like combing the hair over their head in three different directions, right? So we have cells that go in this direction, cells that go in this direction, and cells that go in a vertical direction. So, when I first saw that, I was really blown away. This is a beautiful, almost mathematical arrangement of these cells. And I, instantly I started wondering, how can we guess the function of these cells? What can we do? Well, the obvious thing we can do is let's look at the inputs to these cells. And in particular, I'm going to show you the inputs to the DM3V cell. And so you can see here's the DM3V cell. So let me just back up. That's this is the brain, and this is the right optic lobe. And then this cell is the vertical DM3 cell. And these are three strong inputs to that vertical cell. And these input cells are pixel cells. They are receiving, they're mapped in a one-to-one way to the pixels of the compound eye. And so, once you see this arrangement, this cell is tangential to the optic lobe, and it's going across three of the pixel cells. So once you see this arrangement, then it's pretty easy to make a guess if you've taken introductory neuroscience, right? This definitely reminds one of the picture in the textbooks of the circuit mechanism hypothesized by Hubel and Wiesel in 1962. They said, well, maybe a simple cell is the way it is because it gets input from some number of center surround cells that are arranged in a line, right? So, so what you saw there, the argument then would be that this DM3V cell is going to be orientation selective. It's going to be selective for vertical orientations, bars that are vertical. This Hubel and Wiesel wiring diagram has never really been directly confirmed because of experimental limitations. It's only been tested indirectly in a mammalian visual system, but we can very directly see the connectivity in the fly visual system. And in particular, we can go further. We could say we could map the receptive field or predict the receptive field. There's no physiology, but we have the connectivity. We can predict the receptive field of the DM3 neuron just by the arrangement of those pixels pixel neurons. And so that's what I'm doing right here. So what I've done, these three maps right here are maps of the eye. And what I've done is I've taken three different DM3V cells, and I've mapped their connectivity to this pixel type, which is TM1. And the color here is the number of synapses made by one of these pixel neurons onto the DM3V cell. And so you can see that each one of these DM3V cells has a predicted receptive field, which is vertical. I'll just, for fun, I'll show you two of these cells, right? This is happening all over the visual field. Every DM3V cell is going to have a vertical predicted receptive field. And if we take all of those predicted receptive fields and we align them and we average them, we'll get the average predicted receptive field. And here's what we get. So for several hundred of 400 cells of each type, and we align an average, and you can see something that looks shockingly mathematical. It's as if I made it up, right? But it actually comes from averaging real data. And so the DM3V cell and DM3P and DM3Q have receptive fields, average receptive fields that are aligned along the cardinal orientations of the hexagonal lattice of pixels in the Fly compound eye. Okay, so that's our prediction. These DM3 cells have three different preferred orientations. We can go, the referees asked me to characterize the variability, so that's done here. Now, there were 6 cell types. What about the TMY types? And you can see also here that TMY types, there's 3 of them. This has an oriented dendrite, and this has an oriented dendrite. And then this has a horizontally oriented dendrite. And again, we can map the inputs. These also get inputs from TM1. And so these are the predictive receptive fields of the TMY cell types. So there's 2 trios, and there's a pleasing duality here, right? So these 3 types, their predictive receptive fields are orthogonal to the receptive fields on the left, right? So the TMY4 is horizontal, whereas DM3V is vertical. All right, now that was just the feed-forward connectivity, but there's actually lateral connections between all of these types. And so I'll show that to you now. Oh, okay. Now, first of all, before I do that, I'll just want to make a commentary. If it hasn't occurred to you already, there's a strong correspondence with the idea of a convolutional net. So remember, a convolutional net is an artificial neural net defined on a lattice. Everybody knows about what these are now. They were pioneered by Jan LeCun and so on a long time ago, also Fukushima. And I actually wrote a whole review article about that for Candela and Schwartz. So I've credited them for that for a long time. But everybody knows that for now. But what's surprising is that, because the Fly visual system is on a lattice, it's literally a convolutional net. It's not like some kind of metaphor with a mammalian visual system, right? It's literally a lattice, right? And so neurons are located at lattice points. They're divided into feature maps or channels, and connectivity between 2 neurons is local in space and depends on spatial separation and identities of their feature maps. And this property is known as translation invariance. And so every cell type, when you ask me, I showed you 200 plus cell types, right? And you kind of you might be freaked out by all that diversity, right? But if I tell you that every one of those cell types is a feature map in a convolutional net, oh, then you have some notion of what's going on, right? They're all feature maps. All right, and I showed you the translation invariance also, the approximate translation invariance. Now, I want to say that a cell type is a feature map, but the Fly visual system has some differences from the convolutional that's used in AI. For example, it's likely to be genetically specified with very little influence from learning. I mean, that hasn't been proven, right? But you can dark rear flies and they seem to see just fine. Maybe there's subtle differences that people haven't found yet, but overall, there's a strongly genetically specified. And then the second thing is that the convolutional net in the Fly visual system is recurrent. I'm not, I'm about to tell you about the lateral connections, but there's also top-down connections. All right, so here's a very coarse summary of the lateral interactions just at the population level. Here is, remember there's 6 of these 3 DM3s and 3 TMYs. And here is the interaction between the DM3 types. And here's the interaction between the TMY types. And this is very interesting, right? Because what it says is that DM3 types prefer to connect with other types. They avoid cells of the same type. Now, you might ask functionally, what does that mean? Well, it turns out that it's very highly likely that DM3 is inhibitory based on the neurotransmitter identity. So this is probably cross-orientation inhibition, right? Cells prefer to inhibit cells that have a different orientation preference. And if you look at this, so that's the off-diagonal structure here. Now, if you look at this square, that's got an on-diagonal structure. And TMY cell types are very likely excitatory. So this is going to be iso-orientation excitation. And, these have been, these things have been, cross-orientation inhibition has been hypothesized in the cortex since 1972. Iso-orientation excitation hypothesized since the 80s. But actually, cortical physiologists still argue over what is the reality in the cortex. But through a few lines of code, we can get this for the Fly visual system. A few lines of code. It just took me a few minutes to first make this diagram once we had the Fly Connect done. Okay, now what are these doing? There's an important thing here, which is that iso-orientation. I'm not going to talk about it in detail. We don't have time. This is very rich, but iso-orientation excitation. One question you might ask is that does it look like this or like this, right? Do we see parallel orientation detectors that are side by side, or are they end to end? And what I've shown you here is that they are mostly end to end. So these are TMY cells, and you can see that they're touching each other. They're holding hands, right? And what I'm not showing you here is there's quite a few synapses going in both directions between these cells that are holding hands. Let's look at here's what it looks like. And so there's plenty of places where they touch. That's likely a synapse. You can look at the actual images to be sure, but the morphology seems very likely it's likely to be a synapse. And so, why are they holding hands? Well, there's an old idea, right? So we know that if you look at edges in the real world, the bottom-up input about them might be very noisy or ambiguous, right? So if you look at this scene, you might guess that there's an edge here because of the context, right? Edge detectors here might be firing, and maybe they exert peer pressure on the edge detector here. And of course, illusory contour stimuli are like that, right? So the idea then is it's extremely visual and extremely structural, that this idea of contour completion as being peer pressure between neighboring edge detectors. I mean, literally, these neurons are holding hands. So they're in a position to pull each other up above threshold. Now, I haven't proven that's the case, right? Physiology experiments need to be done, but this is showing you a possible substrate for contour inference. And I want to remind you that Charles Gilbert spent decades trying to investigate this in monkeys. Here's a great example. This square right here is contains the receptive field of a V1 neuron. And if you if you give a stimulus of the preferred orientation inside the receptive field, you get a robust response. If you give flanking stimuli, you get very little response. But if you give the flankers plus the stimulus inside the receptive field, you get a huge response. So he argued there's this facilitating kind of interaction from collinear stimuli. Now, the sad story is that other people, other experimenters, report that these flanking stimuli have a suppressive effect. So the physiologists actually don't agree. It seems to depend on the experiment. And so I just want to, show you that this is a so-called extraclassical receptive field effect. And I want to show you that we can predict extraclassical receptive fields from the connectome also, right? And so I argued that we could predict the classical receptive field by mapping the TM1 input to TMY4. But we could predict extraclassical receptive field by mapping a disynaptic pathway, right? TM1 excites a TMY4, which by holding hands excites another TMY4. And so we can map out the TM1s that are presynaptic to this TMY4 through this kind of pathway. And that's shown right here. This is the TM1s that are monosynaptically giving input to one TMY4 cell. And then these are the TM1 cells that are disynaptically giving input to a TMY4 cell through this lateral interaction. And so you can see that they're at the tips of the classical receptive field. And so again, cortical physiologists can argue about this for decades with no resolution, but we can already look at the connectivity and see the possibility, right? Of this collinear facilitation. On the other hand, we can see why the physiologists have a tough time. And the reason is that I'm showing you only this excitatory pathway, but there's also disynaptic inhibitory pathways that go to TMY4. And so the net effect of what you're going to see here is a combination of excitation and inhibition, and that might make the physiology quite sensitive to exactly how you do the visual stimulus and the experiment. Okay, so, but with the connectivity, the hope is that you constrain this enough that maybe physiology plus anatomy could really figure out how this system works. So this is just the beginning. It's not the end, right? It's just, but the beginning of being able to answer these kinds of questions in a definitive way. All right, so what I've found here then is I've found a new visual subsystem dedicated to form. In the Fly visual system, many people have studied motion, for 75 years. People have studied color more recently. But this seems to be a subsystem that's dedicated to form in the sense that it does the edge detection, it's predicted to do the edge detection as in the way that people imagine for primary visual cortex and also for computer vision. And I don't have time to show it, but downstream, there's another cell type called LC10E that combines input from two TMY cells at this orientation and roughly this spatial relation. It's a little noisy. So there may be a corner detector that's downstream. So that would be like the visual hierarchy that we try to construct with convolutional nets. So making a complex feature out of a simpler feature. And I want to say that recently, this work, this is a preprint out of Tom Clendenin's lab. These are the first published recordings of DM3 and TMY9 cells. This these are the calcium image responses of these cells from a white noise visual stimulus. So this reverse correlation kind of mapping of receptive fields. The pixels are a little different, 7 degrees versus 5 degrees. But you can see right here that there's a great agreement, right? That the size and orientation of these receptive fields, roughly matches the anatomical prediction. So we can say that we were able to, this is one of the great examples of being able to predict a receptive field before a visual neurophysiologist ever does an experiment. Now, all of these more complicated effects that I talked about from lateral interactions, of course, those remain to be investigated. Okay, and I'd like to refer. I don't have time to talk about it, but I'd like to refer you to related work on interneuron diversity. So it turns out that a large number of cell types basically many cell types in the fly visual system are analogous to normalizers in convolutional nets. It's well known now that normalizers were already there in the early work of Fukushima, but it's well known now that convolutional nets perform much better if every feature map is normalized. And in fact, you can find analogous but somewhat different normalizations in the fly visual system. And then this preprint is about the problem of biophysics, right? So is the point neuron approximation valid? And I show that there are certain cell types, some of these interneuron types actually probably have multiple outputs. They don't just have a single output. So it's some kind of biophysics probably has to be taken into account. All right, and so I'd like to wrap up and thank my collaborators, Malamurti and the FlyWire Consortium, and also the cell typing efforts of Ari Matzlia and CJ Yu, and also a team of annotators.